So, did you get the gap, research gap, why the studies they are doing? So basically, when you take any medicine, they have side effects. Okay, so that is the basic assumption or reality. So for cardiac arrest or cardiac failure, the one agency that SHIPA, they have mentioned that some drugs has several side effects in our heart. So if circadian rhythm is affecting particular cellular activities, and if you apply to drugs to avoid that activity, the drugs will cause another problem. So that is the criteria to assess the drugs for the cardiac heart failure. Okay. The SHIPA has evaluated that 28 drugs are there. They cause a cardiac problem if you consume them. So now in this paper, they have taken 11 drugs and do a computational study and to investigate how these drugs are modulating the cardiac property by studying the cellular properties. So this is the abstract for this paper. So we have divided the objective in two types. So first we will be familiar with the basic cardiac electrophysiological terms, that means electrical activities in the heart, what we will study and what they have investigated. Then we will see how the simulation or the computational model had established some other parameters, set of parameters to study this drug that is not happening in the experimental. And the content of the presentation is in two parts. First is basic electrophysiology and basic computational modeling related to electrophysiology. And second is related to this paper results, that is dosing time of the impacts in cardiac electrophysiology. So let's go to first section. So basic cardiac electrophysiology and computational modeling. So in this section we will see how heart is functioning and how our heart is related to electrical activities, our heart is related to mechanical activities, and the electrical activities are distributed different region of the heart, and how the electrical activities are modulated by cellular activity, and how cellular activity are modulated by the circadian rhythms, and how different drugs modulate circadian rhythm and electrical activity in heart. Okay. And second part is how the mathematical establishment or the computational flow happens from single cell, then tissue, then organ. The heart is as a whole organ. How different equations are involved in for single cell, then tissue level, then organ level. And what this paper will try to understand at cellular level. Okay. So this slide is related to basic cardiac electrophysiology. So here you can see one picture of heart and some graphs, and this I am going to explain all these graphs. So the basic function heart to pump the blood, first to collect the blood from the upper side and the lower side. So you can see the heart has two sections. One is, this is the right side heart, this is left side heart. Okay, so this heart collect the blood, that is dirty blood, from the upper and lower part of the body. Then it will go to lungs. Lungs purify the blood. Then again it comes to the left heart. Then it again transport to upper part and lower part of the body. So now in this heart we have four chambers: ventricle and the atria. Okay, so to pump the blood from one chamber to another chamber, we need contraction. Okay, so here is the contraction happening from this side to this side, and then from this side to this side. So contraction is a mechanical term. So to generate the force, we need calcium. So calcium is stored inside the cell. The calcium release profile is happening because of some consequent electrical activities. These are known as the cardiac electrophysiology. Okay, so we are all familiar with the lower figure. Can you say what is the meaning of lower figure? This figure. So many movies we have seen this figure. This is basic electrical term in cardiac electrophysiology of cardiac. So when you go to clinic, they check all these things. So this is ECG. Have you know ECG? Electrocardiography. So ECG have like in movie you have seen the tick 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 tick. So many. Okay, so if it is not stand still, that means you will be the person will be dead. Okay, so this is ECG, and you know where is how the ECG is recorded. You know the electrodes here, 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 here. That is 32 electrodes, 16 electrodes above the skin, the circular electrode. Okay, so suppose I have 8 or 16 electrode here, so that is external to my body. Okay, so it accumulates all the electrical activities from the heart and then collected all these electrical activities and generate this type of graph. This is ECG graph. So ECG you can see, so the letters are P, Q, R, S, T. Okay, so these are the particular time of the ECG, something happening inside the heart. So when we say the heart is, we have some particular beats of how. Do you know how many beats is healthy for the heart? So we have a specific range, like below 100 it is good, above 100 is bad. So this particular we in ECG machine we saw the we see how the graph is running and per minute how many peak is happening. So that is calculation of ECG beat. So now the cardiologist or doctor can see the how our abnormal ECG is happening and because of which modulating property. We can say that if the duration, here you can see the all the duration can be changed, that will cause the problem, and the peak can be changed, that will cause the problem. Okay, so overall the ECG is most important factor for the cardiologist to check the abnormality of heart cardiac arrest, heart failure like this. And this ECG is happening because of individual electrical activity in this heart. Okay, so when I say individual electrical activities, so the ECG is because of external electrode above the body. Okay, so that is external. ECG is injecting the electrode inside the cell, the cellular electrical behavior. So here you can see 1, 2, 3, 4, 5 couple of electrical activities. These are actual electrical activities from the cell. Okay, from which cell you can see the different names are there: SA node, atrial node, AP node, all these nodes. Okay, so each location can generate one spike, that is action potential, and because of combination of all this spike, we'll get ECG. And the ECG is the first property to check for the cardiac arrest or cardiac failure. If ECG abnormal, that means some of these electrical activities are abnormal. Okay, so in research, people study one of this spike or action potential to find out the actual problem with ECG. Okay, so in this electrical activities, you can say there is a steady state period, then there is certain rise, then falling, then again there is going to steady state period. So this is known as action potential. This is rise, this is falling. In electrical term, you can say the deceleration, receleration, and the resting membrane potential. Okay, so if the rise time is changing, that will cause problem. If the falling time is creating a problem, that will cause problem to ECG, and the duration is different, that will also create problem with the ECG. That means indirectly to the heart function, or it will cause cardiac arrest or heart failure. Okay, so here I have shown that this is VAP is known as ventricular action potential, and the ventricular action potential is generated from the ventricle. So that is the most important part of the cardiac arrest or heart failure. So now most of the researchers are studying how this ventricular action potential valve is changing the ECG pattern. So now we are not going to see the ECG pattern, but we'll see the VAP, what is happening to VAP for particular drugs. Okay, that is related to circadian rhythm. Okay, so I have then appointed some terms. So in cardiac electrophysiology, you know arrhythmia. Arrhythmia is any abnormal electrical activities or any abnormal electrical activity ECG known as arrhythmia. Okay, so if you take drugs to prevent arrhythmia, that will be antiarrhythmia. So this is antiarrhythmic drug. So any drugs that will solve the arrhythmia problem, that is antiarrhythmic. So if drugs causes another problem, if due to drugs, if our heart goes run faster, that is pro-arrhythmic. Okay, so some drugs are pro-arrhythmia. Okay, so if you take the drugs, so it will cause one problem. It will solve one problem, but cause another problem that again heart will run faster. So that is pro-arrhythmic. So now next is tachycardia or bradycardia. Tachycardia is more speed of the, normally if your heart beats is more than 100 beats, that is tachycardia. If it's too slow, that is bradycardia. So tachycardia and bradycardia are problem for the heart. Okay. So and next is the most important and most fatal or most dangerous tachycardia is TDP, that is towards the points. So this is a fatal type ventricular tachycardia that is happening due to ventricular action potential generation, and it will prolong the QT duration. Okay, so you can see the ECG Q and T. If the duration is takes more time, so that will create a problem. So that is known as TDP. Okay, so in this simulation they have evaluated this TDP. So if you can evaluate the TTP, we can evaluate the drugs, and we can simulate all the drugs, how they are causing the TTP. Okay. So the main theme of the slide is that if you explore the cardiac electrophysiological through ion channels or cellular or tissue level dynamics, we can explore new drugs, or we can simulate the drugs already we are using for the cardiac safety. So next we'll go to computational modeling. So how the computational modeling happening to cardiac electrophysiology from cell to tissue and organ. So computational modeling cardiac electrophysiology provides a powerful platform. So for ion channel, cellular and tissue level investigation. So can you even say what is the problem in getting recording from the heart cell inside the heart cell? So in experiment, so heart is like a one network. So if you inject electrode in one cell, it can cause or it can break another cell. So the experimental, maybe mouse or maybe guinea pig or maybe human, very difficult to get the actual recording from the heart. So again, one drug causes multiple cellular activity. So here cellular computational platform is like in silico electrophysiological setup. So you have a cell, you have simulation setup, you can inject current, you can get recording and evaluate the output. Okay. So here you can see one figure. So this figure has three parts. You can see here is ion channel model, that is the cell subcellular part, then cell model, that is the cellular part, and cell to organ model, that is the heart. Okay. So when you go for computational modeling, the first part is to interpret everything in mathematical equations. Okay, so here is the subcellular activity or the ion channel model. So here you can see some equations. We'll go all equations later. So you can see the one single ion channels, their equations, and the ion channel output, that is a current. This is inward because the inward current, positive current, so this is current. So single ion channel, the respective equations to evaluate the ion channel and the single current. Okay. So now we have different ion channels that will form a cell. So now we have a single cell. Okay. From combination of ion channels and other cellular component, it's a cell model. So here you can say the cell. The outside is the outside of cell, that is blood related, and inside is the cytosol, that is inside the cell. So the cell can communicate. You can see the some ion channels across the cell membrane, that is not membrane. The membrane coordinate the flow of ions. So when you take more salt, it will cause more contraction. So if you take more potassium, it will cause another problem. More calcium, it will cause another problem, because the imbalance or diffusion or the property here, the inside concentration and outside concentration of the ions are changing. So that will cause the electrical activity inside the cell. So here you can see the action membrane potential that we have seen the earlier slide. So that means all the cellular labor were generating the electrical activities in terms of spike or action potential. And this action potential is propagated, the cell is connected to another cell, propagated to other cell, and they coordinated electrical activity and coordinate contraction, that is the organ. So now here for cellular level, the total current equation is like this. Again, we will go later for this equation. And all these cells are connected to make an organ. So how to study the organ level study, the propagation of signal. So these are the equations. So this part we are not going to cover, only this cellular model. We'll go for the single cellular model. Okay. Then how to interpret all these things in a mathematical schematic diagram. So here we'll go to electrical interpretation of ion channels in the cell membrane. So single ion channels will go first. You can see the single ion channel. This is a cartoon. So here you can see this is the outside of the cell or ion channel. This is inside of the ion channel. Okay. So let's assume the ion channel is the door. The other side is the outside. The classroom is the inside. Okay. So if some person is coming, that is the flow of ion inside the cell. Let's say door is half closed, the door is quarterly closed, door is fully closed, and door is fully open. So we have four options, three options, that fully closed, fully open, half closed, half open. Okay. So then number of person will be allowed. It will vary. So if it's fully open, more person can come. If it's fully closed, nobody can come. So if it's half open, then one person can come. So that is the door of this. This is the ion channel door. You can see. So here is the door. And the function of door, the function of closing the door is evaluated by the potential, or let's say by external force. Okay, if someone is there, that will open the door or close the door, that is the external force. So anyway, the opening of this door is a probability function. So it is full open, this is 1, if fully closed 0. That means the current, the equation you can see, the current depends upon this opening and closing of the door. Okay, so these close and opens doors are operated by some mathematical option. We'll see here how this mathematical happening. Okay, so an electrical equivalent, this is the electrical battery, this is a cell, single ion channel, this is the current is coming, this is a capacitor, that is charge is accumulating. So this is a single cell equivalent electrical pattern. Okay, so now for a cell, if you go to this figure, this is combination of multiple ion channels. So now we'll see how the multiple ion channels are happening. So multiple ion channels, like this is a cell membrane. So we have several ion channels are there like this. All ion channels can be assumed like a door. It will allow the person a specific ions, calcium, potassium or sodium, but due to external parameter, maybe voltage, maybe calcium, maybe temperature, maybe anything. So this is the ion channels and this is the exhaustive cell membrane. And what is the electrical equivalent? That is known as a parallel conductance model. So all cell ion channels are represented by a variable resistance, by a battery, and the current. Okay. So when you say the cell model current, all currents are summed up. Okay. So in this computational model, so they have taken the total current from a single cell to simulate the drugs. Okay. So now we'll go for the equations and how the drugs is modifying the equations. Okay. So governing equation, first is single ion channels. So like I have mentioned the door analogy. So here is the equation. You can see the I ion, maybe calcium, maybe potassium, maybe potassium, G, M, H, and Vm minus E ion. Okay. So E ion is a nonce potential. So that is the individual property of ion channels, and Vm is the membrane potential. So now this voltage difference. So if you this voltage difference governs the current, then this M and S. Let's assume that M is a parameter that is governing the opening and closing of the door. So that means that will affect the current, and also H is another parameter that is also modulating the opening and closing of gate. That is inactivation parameter that will also governing the ion channel. And G ion is the how many persons are coming. More person, more current. Okay, so more G, more current. So G is the conductance parameter that regulates the ion. Okay, so in all our next slides, this is the governing equation. So we will see how the current is changing with respect to G. Okay, so for individual ion channel, we have couple of ion channels, 4 or 5 ion channels. We will say this current will change because G is changing. G maximum, maximum conductance. G minimum, minimum conductance. And we will see that how G is governing the ion channel. So this is the formalism is known as Hoskin-Huxley formulation. Okay, so there are several methods to form the ion channels equation. I have only taken for this HS formulation because the this paper has established on this formulation. Okay, We can say Markov model, we can say multiple state, we can say not quantum mechanics, everything now governing the ion channel parameter, but only we are taking deterministic HS formulation. Okay. So next is, so here I have mentioned that G ion is conductance, that is the most important parameter. So the M's and are governed by some differential equations. So like time constant, the steady state value and the instantaneous value. Then H is also governed by differential equations or ODEs. So next is GSK equation for calcium.
Oh, sorry. Could you explain what is the difference between Mx and Hy?
Okay, so here M is known as the activation parameter and H is known as inactivation parameter. So I have just told you that the door is governed by one person. Okay, that if person is closing the door or varying the closeness of door, that is the activating it. Okay, so now imagine two persons are there. Okay, so one is activating, that is okay, but if activate the door, you cannot inactivate instantaneously. There is some latency, there is a time. So that is caused by the another parameter. He will check how much delay he will get to the door. Okay. So that means both persons are modulating the opening of the door. So both persons have transient value, steady state value. They are also modulated by the membrane potential, temperature, or any other parameters. So that's why this M and H are important. Okay. But for all ion channels, M and H are not essential. For example, for potassium channel, only M, only activation parameter. Okay. So see here the parameter. Now GSK equation is gold when Huskin equation when the equations are not linear or there are some diffusion process. So imagine you have the much calcium concentration here and less calcium concentration here. So due to diffusion, calcium will flow from higher concentration to lower concentration, where you can say some Hales coefficient, then some other equation coming. So here in for calcium we have known as the ICA is calcium current is governments this equation. The PCA is important. So PCA is known as permeance. Okay. Permeance is for the calcium. So this is for diffusion. Okay. So the permeance depends upon calcium concentrations. So you can see the calcium concentration Ca, I, Ca 0, 0, 0, 0, no CaO. This is outside concentration. This is inside concentration. These are coming into equation. Okay. So now for rest of our equation or rest of our slides, just remember that we are going to change the G conductance and permeance for the calculation of the current. Okay. So if you go for the multiple ion channels, that is just formation of single cell. So here all ion channels are there. Then the total current will be evaluated. The net current will be evaluated by the summation of all the ion channel current. Okay, ion 1, ion 2, and ion 2 like this. So the I net is the most important parameter to check the capability of one particular drugs. How the paper have evaluated drugs because of this I net. Okay, so they have evaluated I net. Then they say that this current is changing. That means drugs is modulating. So we have a safety parameter for that drugs. Okay. So now I net depends upon individual ion channel current. Then that means individual ion channel conductance or permeance can change the I net. Then if drugs can change the conductance and permeance by a particular ion channel, that will change the I net. That means the total property changing. So here we will evaluate the drugs, how the drugs is modulating the G and P and the I net. Okay. So now we will go for a computational setup. So here is the computational setup. So as I have mentioned, that is we are going for single cell study. So let's imagine this is a single cell. Okay. All ion channels are there. This is a single cell cardiac cell model. Okay. So how to know this system is working? So what happens in experiment? We inject some, the defibrillator in bus station and train station. There is just some device in defibrillator. That will keep when people are suffering from cardiac arrest or cardiac failure, they just put that machine. So that will inject one heavy current. Okay. So if I inject a current to heart will function and it will generate the voltage or action potential. So in our software setup or simulation setup, here is the cell model. We will inject current and we will see how the outputs are there. By evaluating all the outputs, we can say that drugs is changing the output. So we can say the drug has some safety index. Okay. So here is the input as current, stimulus, step current. Okay. You have some duration and some amplitude. Then when you inject current to the cell model, it will generate some action potential. Okay, by evaluating the action potential, here I have set up action potential. You can see the change in the rising curve, change in the falling curve, changing the resting membrane from. So all these shapes are modulating the ECG. Okay, and ECG defined if if the person is going for cardiac failure or cardiac arrest. Okay, so now in this paper we have one cell model. We'll inject current. We'll see the output. So now in the model, cardiac modeling has started in 1950. By people from Oxford and US, they have started. So they have first tried to do the mathematical interpretation of cardiac model. Okay. Then we have seen the first slide. Several places are generating several electrical properties. So several researchers are model the cardiac in different point. So we have several models. So now when we go for one cardiac model in computational modeling, we'll pick one model, best model or latest model. Okay, we'll start from that model. Okay, so similarly, this paper has evaluated their computational study from one particular model. We'll see in the next slide. Okay.
Excuse me. So, I'm not so familiar with HH model. So, could you explain the M and H, the role of M and H, and is it the 3/1/2 is a parameter?
Yes, parameter. Okay. So in experiment, okay. So in experiment, you see that M is activation parameter. Okay. So activation means it is supporting the conduction. So it is more. It is like a. Okay. So this is my x-axis. Okay. So this is my y-axis. Okay. So when I say the door is closed and door is open, the probability is open means fully open means 1. The fully closed means 0. So let's say this is the door opening and door closing. So this is 1 and this is 0. This is the function. Okay. So when we say the function is function of membrane potential, x-axis is the voltage. Okay. Okay. So that means the probability of this one M that is activating, that is opening, and the S which is inactivating, resisting. So both are probability 0 and 1, but they are the function of the voltage. Now for the M who is trying to close, trying to open, initially it is at 0 level. Okay. So when voltage is increasing, it will go like this. Okay. This is the steady state. Okay. So H is inactivating, so initially it is. Okay. So now here we have set up voltage. So there is a window. In window, this is 0, this is 1. We have some mid value 0.5. Okay, 0.5. This 0.5 has some voltage. This is VR. Okay. So that means for this sigmoid curve, this is M, this is H. So we have M half, we have H half. This is steady state value for a particular voltage. This is the steady state value for a particular voltage. So now this M and H are function of V. For a simple ion channel. But if your temperature, your calcium concentration, for example, the next equation is a complex equation. Okay. So for the simple equation, M H are the function of the voltage. We have V half, M half, and MS. So when we say that there is no H, that means we have only one M. Okay. So some ion channels have only one M activation parameter. So they do not have any H. Okay. For example, potassium ion, so they have only M. And for sodium, they have both M and H. Okay. So now M and H are function of voltage and maybe some other parameters. Okay.
Then the general version has the large T is the temperature.
Yes. So in this figure, you can see F is Fahrenheit, V is the membrane potential, R is gas constant, and T is the temperature. So actually, the term A, V, and Z is association constant. Z is 2 for calcium, and for 1 is for sodium. Okay. So only one thing here, conductance is a matter of investigation. Here, permeance is a matter of investigation. So when you say what is the meaning of conductance permeance, just say the door has some opening space. it is okay, door is fully open. So let's say the door is wide like this, the door is wide like this. So here is the conductance and the permeance difference. Opening is more, but 1% is coming. Opening is less space, but 4% is coming. So that means the current is changing with respect to conductance and permeance. Okay.
Okay. I'm confused, but so HA formulation and JH equation saying all to use in the model. Two of them are used in the model.
Yeah. So some ion channels. So in 1950s, when they have started interpreting mathematical equation, so they have done actually from experiment. They see how the voltage is changing this opening probability. Then they have said this B half and for M and H. Okay. So some ion channels. For example, this is my T. This time. Okay. So I have a particular ion channel. Let's say this figure. We have a potassium ion channels. Okay. So I am applying voltage. So this is my current. Okay. This is current. Current unit is ampere. Let's say ampere. Okay. This is a potassium channel. This symbol is IK. This is time. The axis is time. With respect to time, I will say how the current is happening. So if say for 0 there is no current. For 1, this is 0, less 1, this is 2 like this. So for 1 like this, for 2 like this, for 3 like this, it's happening. Okay. So here we see that there is no inactivation. After the voltage is increasing, this is a steady state value. It is not coming down. Okay. So some ion channels current profile is like this. More voltage, more current. So it is like this. That is only M matters. Okay. And some currents are like this. Let's say again this is T. Let's say sodium current. Sodium symbol is INA. Okay. This is 0. Okay. Let's start 0 millivolt or 0 volts. There is no current. Or let's say one particular voltage, let's 50 millivolt I am applying some channel. The current is like this. Okay. So that means this factor is determined by the M. We can say activation parameter. But what is modulating this parameter? That is inactivation property. Okay. So this is the profile of M and S. So initially in potassium channel only M was coming. When with the sodium channel, H is coming. And here you can say there is a window. Okay. So when says threshold of the ion channels, so here you can define the at which voltage this ion channel is opening. Okay.
And then when the GHK equation is used, GHK equation for calcium.
Also, GHK equation is complex. The HS formula is very simple. Only OD is. So when we have calcium function, so let's say the door function. Okay, so the door 2% are there, but only they are not opening to the person. They are also opening to suppose cleaning person who is coming. They are not sensitive to only the students or lab member of the lab. They are also sensitive to other cleaning staff, emergency staff. So that means other staff can trigger to open the door. Okay. So when multiple parameters are coming, then that is GSK equation. So let's imagine calcium is another factor which is also modulating the ion channels or calcium ion channels. So here we are going for the permeability. Okay, the PCA function is depends upon this equation.
So GH equation for it is not for some kind of specific receptor, but
Yes,
the general permeability.
So this is the two simplest equation I have included. So the more ion channels, for example, for our lab, they have. So here, this is not MS. If you go for stochastic or Markov chain model, the M has like 16 step. Opening 0, opening 1, opening 2 like this, closing has multiple steps. So that is more complex. Okay. So with more experimental leisure, you can go for more computational complex computation problem. Okay. So here only two equations, HS and GSK, because these equations are the governing equation for their paper. Okay.
So is that shadow is related to the communication? The question.
Yes.
Yeah, communication. So how the cells are communicating with each other.
Okay. So this is a cell 1. Okay. So this is door, this is ion channel. Ion channels are there inside the cell. Okay. The cell generating action potential. Okay. So now your question is how cell are communicating. Okay. So organ level is heart, level is heart. The cells are integrated to make the tissue, then organ. The another cell is connected is like this. Okay. So there is a gap junction. Gap junction is the communication channel between two cells. So in gap junction, we can communicate electrical signal. Also some gap junctions are there where communicate the calcium signal. The all cells has inside store. This is calcium stores. Okay. So because of the cellular function, calcium releasing. So normally molar is the unit for calcium. If you are in normal, it is 1 nanomolar. So it goes for the 20 nanomolar. This cell will contract. So what will eventually do? We need contraction. So we need more calcium here. So for more calcium, we need calcium release from this body. So this calcium release is function of all these ion channels. Okay. So now the electrical activity is intended to release calcium from here, and also communicating to this cell. So for coordination, this cell should send signal to this cell, then another cell. So this is now network. Okay, so when we say puchu to pump the blood, that means coordinated contraction by the releasing the more calcium. Okay, so here is the cell communication happening. But for individuals ion channels is happening every cell. Here ion channels are there, here ion channels are there. Okay, so only have taken only one side. So finally we have a setup. We will see inject current. I will see action potential. Now we will go for the result part of this paper. So how the dosing time, how the different, you have 24 hours, 12-hour light and 12-hour darkness. How this 24 hours pattern of taking medicine is changing the cardiac electrical activities. And again, it depends upon the different type of drugs. That is already defined. These are the heart anti-arrhythmic drugs. Okay. Okay. So what is their method? We know the simple method from our previous slides. We'll go how they have used. So modeling approach is here. You can see. So okay, this SIPA is one organization coming. They have evaluated that we have 28 drugs. These are pro-arrhythmic. Okay. So from this that SIPA mentioned drugs, they have evaluated this section. Here you can see. Can you relate this paragraph to our previous discussion? The conductance and the permeance. Okay, so they have checked. For example, you have a 24-hour cycle. You can check all the conductance. So they have a conductance cycle for 24 hours. If you check the permeance, they have a permeance cycle for 24 hours. So just take one drugs, check the pattern for conductance, check the permeance with the conductance. They have done these things. That is the modeling approach. Okay. So what is the governing equation? I have already mentioned that G is the conductance. This is sodium channel. So we have a minimum conductance, a maximum conductance. This is a cycle. If you check for 24 hours, this is a cycle. So if this pattern is maximum, that means sin theta 1, then this will be maximum value. So that depends upon the time of the which time, JT0, JT6, JT12, JT18, JT24. So if a particular conductance is maximum at JT0, we will get maximum conduction at that time. Okay. JNA means more current. More current means that ion channel is functioning. Okay. So next is PCA. This is for calcium channel. Again they have a cycle. So here the peak time is JT3. You can see how this 12 is 90 degree. Okay. Then next is JKR. They have taken only four ion channels: Na sodium, calcium, potassium, and total. This is outer transient current. Okay. So 1, 2, 3. Another is the outer transient current. So here we have the governing equations. So they have taken from their model. So now for individual drugs, they have evaluated the GNA, PCA, GKR, GTR. Then they have done for bar graph, plot graph, evaluated how the sensitive. Then calculate for the results. Okay. So next is drug simulation setup. So we have 28 drugs. The SIPA has evaluated 28 drugs. They have taken only 11 drugs. Why? Because before going for any simulation, we need to validate against the experiment. So in the previous model, they have taken another base model. So here you can see the model, Hararubi model. So this model they have evaluated 11 drugs. So now as they are taking this model, they are also evaluated 11 drugs. Okay. About total 28 drugs are there. Okay. Next is how to measure the safety. So they have evaluated the current, then they have integrated current the 24-hour cycle, the full cycle, then the more current, more safety, less current, less safety. Then what is the governing equation? This is the governing equation. Okay. So this is the last slides of the method. Next we will go for the only results. Okay. So anything?
Yeah. So in the equations there, so we said that the peak occurs at the T0. Does that mean the T0 means that we need to plug in 0 at T?
Yeah, T is equal to 0. Yeah.
Then why the phase is 28 there?
But they have done some, fitting, okay, to fit the mathematical equation to experimental one.
So what the peak occurs means that the sinusoidal
Yes.
peak occurs at
Yeah.
T0.
So you have to validate with the experiment, they have done this peak,
Okay.
but some experimental and computational modeling are not fitting, but closely similar, so they have taken some other parameter to change the offset. Okay.
So when you plug in T equal to 0 there, then the sine value becomes not pi half. So that does not mean that we get the peak value at T equal to 0. Maybe there's another phase term, or the phase value is something wrong.
The first one.
Yeah, so if you plug in T 0 to T there, the sine value is not 1.
Yeah, so it will be maximum value, I think. So minus, so it is pi by 12.
Yeah, maybe.
Yeah,
That should be.
yeah. So and plus 1 is there to the effect not to negative value. So they have taken this for to calculate maximum value.
And is there okay.
So for the cycle, what is the maximum value of G? What is the minimum value of G? And when the maximum value is happening in the 24 drugs, for a particular drugs. So all drugs will not be like a sinusoidal function, but we should get the maximum value at a particular time and minimum value at a particular time. Just to compare with the experimental result. Okay. So that you can see the next figure, how they have validated the experimental one.
So I have another question. Time scale of electrophysiological model is quite small. It's millisecond activity. So now we deal with the day time scale.
So the circadian rhythm have two parts. One is from this central, one is the local. So in local ion channel they have one clock that is changing the conductance in 24 hours. Okay. Okay. So at a particular this is JT0. So less JT12 for a one phase, light phase. Okay. So in this 12 hours, the conductance is maximum at a particular time for a particular drugs and minimum for a particular time. So that is the evaluation method. But when this time, so that original time t is not dependent on is not the time scale of this zt. Okay, so that t is what is happening. Suppose you are taking these drugs, so that drugs will bind some receptor, then will change the conductance. So that conductance will change the current. Okay.
Okay. So this is basically simulation data.
This is
simulation study.
Yeah, their method for simulation, and they have validated the experimental one first. So first they have validated the experimental one if these functions are
Yes.
actually
So
correct.
then that means you should generate the simulated data.
Yes,
in the
yes.
time.
Okay.
But that simulation model, so the spike occurred within the millisecond, right?
Yes.
In millisecond. So you need to collect all the time series data so that the size of the data should be very, very large. So I wonder how they deal with that super large size data.
So in simulation, they are not going to generate for the all this time. In experimental, so they have suppose in experimental a particular, suppose let's say we have taking a nafidafine, okay? This is a drug of the L-type channel. From experiment, we know that the nafidafine effects has maximum at 12 hours or 6 hours, okay? And minimum at suppose 3 hours from experiment, okay? For simulation, we will take this value 3, we will take this value 6, okay, this is minimum, this is maximum. Now we will have check conductance to obey this pattern, okay. So simulation are not going to take this data, okay.
Thank you.
Okay, so now we'll go to direct to all the results. So first the question like how they have validated, let's say, okay, so here can you say which one is experiment, which one is simulation? So which color is simulation, which color is,
Red is
red is
simulation.
simulation.
okay, and can you say which current is? So, Na is sodium, so this is sodium current. So they have evaluated 4 current, okay. So this is the sodium current. So now, so what is the conclusion from this figure, from the simulation point of view? Is simulation is good, or our equations are good?
No.
No. So here is the point. This is the experimental point. Here is the point. Here is the point. Okay. No clear reading. Okay. Higher at JT0 than JT12. Okay. So but now closing. So what is the modeling inside? Simulating contrast reflects the time of day difference.
Only just the fact that
So at 0
and
0 maximum.
Okay.
Okay. So what is the? Can you say the x-axis, y-axis here? SCM. This is protein. So the experimental have done to number of proteins. Okay. In simulation, we have done the conductance. Okay. So there is no direct comparison between conductance and conductance. Okay. So they are imagining that number of higher protein means higher conductance. Okay. So but the pattern, the simulation, the idea is that if at 0 it is maximum, at 0 it is maximum, at 12 it is it's like lower, 12 is lower. That means ZT0 has higher value than ZT12. Okay. This is for calcium current. So here you can see the pattern is like this, but as we are going for simulation, so our equation simulating like this, but is closely matching. Okay. Again, this is protein, this is conductor, this is permeance. Okay. they have mentioned the clear rhythm is pattern is matching. Okay. Then potassium current. Okay. This is one particular potassium ion channel, HEFG, most important to function. Then this is total outer current. So here when they have one segment, so they have compared like this. Okay.
So, for the sinusoidal model, so when their parameter, for example, their E max values, will fit it to this data? Or
Yes, this experimental data.
Okay.
Okay, so 4 ion channels they have validated from the simulation. Now they will go for their additional result that will be helpful to cardiac SIPA community. Okay, so we'll evaluate the 11 drugs. So they are timing, their maximum conductance, minimum conductance, total current, then the safety index in next all figures. Okay, so the TDP I have already.
Is it okay to fit the data, fit the model by using just 2 data, like in the previous slide? The below one has only 2 data.
Yes, they have taken only 2 data, so they have shown that this person is matching. Okay, so now they have only 28 drugs are evaluated by the SIPA community. So they have taken only 11. Here they have taken only 4. Why? Because of the unavailability of experimental data. So now we will go for some bar graph. So here you can see the TDP of all 11 drugs. So another most important topic of this simulation, suppose we have said the drugs are classified according to high sensitivity, low sensitivity and medium sensitivity. So can you say a high sensitivity drugs, if is taken in morning, will be less dangerous to a low sensitivity drugs which will be taken at morning? So that study will evaluate. Okay, how the 24 pattern is modulating the sensitivity and the drugs in a cardiac pattern, cardiac circadian pattern. Okay. So here you can say that Q net is the total charge. If you remember the first equation, it is the total current. Q net is the charge. Q is the charge. Charge depends upon current. We have evaluated the total current. So how the Q net higher is safer. You can see the higher is safer, and the more safe the drugs are more safe, and the low value is less safety. So these drugs are more sensitivity for the cardiac arrest. Okay. So what is the insight? The TDP risk is not constant, but it depends upon the 24-hour cycle. Here is a 24-hour cycle. That means the one particular drugs, the cycle is changing. That means at a particular time it will be risky. At a particular time it will be less risky. Okay. So this is the relative Q net. You can say the control to up to trace. Again, we go for relative Q net. The pattern is changing because in first equation also it is the difference between the minimum and maximum. So again this pattern is changing for the 11 drugs. So next we will go for the bar graph. How the minimum to maximum is changing for different drugs. So here you can say the window is minimum for a particular drugs, and here the window is maximum for a particular drugs. So this is bar for safety gain. Then another figure is the plot. So here you can see in this figure, can you get anything from this figure? This field, like I'm giving this statement. So what is this zone 0 and what the field and how this open circle? So this field means this is safety zone. Okay. So these are in open means they are not in safety zone, these drugs. So they have classified all 11 drugs according to the time, conductance in a plot, that how many drugs are in safety zone and how many drugs are not high risk zone. Okay.
Sorry. So I missed the explanation of Q net. What is Q?
So Q net is okay. So net is plus total. Q is charge. Okay. So this is integration of current for a particular time. Okay. So let's say full cycle. that the Q net is that means integration of I net. So I net is total current. Okay. So for a particular cycle, let's 0 to maybe 24 maybe action potential, you can evaluate.
So then QNet was calculated from the model simulation
Model
of
simulation.
single cell.
Everything is single cell.
Okay.
So the total studies for single cell.
And how these drugs modify the, for example, conductance and permeance was already known
From experiment.
from experiment.
So past data from experiment, they have validated it. So you can see the difference in equation you asked that the burst to just to match the experimental data, they have modeled the equations.
Okay.
Now they have a model is ready. So next is to evaluate individual drugs by 24 hours.
So then when they do the experiment to measure the parameter or conductance things, did they consider circadian or did they fix the time of day?
Yeah, from experimental the data is according to circadian rhythm.
Okay.
Okay.
Oh.
So let's we have 4 drugs, okay? Drugs 1, drugs 2, drugs, and let's say to 1 drugs, okay? So in experimentally they have value, so let's JT 0 and JT 12 is the 2 point where the drugs is minimum and maximum, okay? They have modeled the computational simulation and they closely generated this one. That this equation is simulating this curve and closely matching. So our system is validated. Okay. From experiment, the 24-hour cycle of D1 is not generated. How the drugs D1 is changing the conductance value for 24 hours continuously. That is possible in simulation.
Okay.
Because you have equation or model, just change the time, you can get one value. Okay?
Does this simulation uses the previous sinusoidal trajectory?
Yes.
And the sinusoidal trajectory, I'm a little bit confused about that. So the sinusoidal trajectory was obtained without drop. With drop?
Sinusoidal trajectory for simulation.
Yeah.
So they have used the equation to generate the sinusoidal trajectory.
Okay.
So they have compared with experiment for maximum and minimum to validate the equation. Okay?
To validate
Yeah.
the equation.
So once the experimental and simulation data are validated, they said our system is good. Okay. Okay. So now we go to generate the drugs for 24 hours. How the GNA is changing for 24 hours, 0 to 24. So this pattern is not evaluated by the experiment.
Okay.
Okay. So this is the additional simulation advantage they are doing now. So from the GNA, they calculate now INA. Okay?
Then we integrate it.
Yeah. From INA, they will go for INET. From INET, they will go for QNET.
Okay.
Okay? Now for QNET, they have the graph. Okay, the QNET is behaving like this. Okay. So from QNET, they go for relative QNET. How the window is there. Okay, minimum QNET to maximum QNET. Okay. Then they have evaluated the safety index from this QNET graph. But all these 11 drugs. Okay.
Okay.
Okay. So now, so here you can see this figure. Individual ion channel, like you have mentioned how they have evaluated. So you have taken 4 ion channel, the membrane potential, how different the drugs are changing the pattern. So here one drug is random pattern. Okay, that means how the J12 and J15 are changing the pattern. So one is for particular time and the green is for another particular time. That means for particular drugs, that is evaluating the current, voltage and everything due to circadian pattern. So this is for inward current, sodium current. Then this is for calcium current. Where is calcium current? Here is calcium current. There's not difference, okay. But then potassium current, then the total outer current, then the total summation of current. So the idea of this figure, so here you can see this is the action potential, okay. So what I have mentioned in the first slides, this action potential is regulating the ECG. If this changing, that ECG will change, that will cause cardiac arrest. And this change of this action potential is happening because the currents are changing. And drugs are changing the current, the action potential, then the ECG, okay. This is the idea. Next, they have done another figure. Again, two drugs are evaluated. How two drugs are changing in different time pattern. So one is the membrane potential. You can see how it's changing the different drugs. Then current, total current. I net is total current. Then sodium current. You can see the sodium current here. Then potassium current. Here you can see the IK1. Then total outer current. Here is T0. Here is T0. Then, so what is happening? Again, same. The membrane potential is changing because the currents are changing with respect to different cycle at different timing, and different drugs have different effect. Okay. So why they have taken these two drugs? Because one is higher risk, one is lower risk. The lower risk drugs and higher risk drugs are also changing the pattern with respect to different time. So that is the most important finding from this study. So next, simulated TDP, again for the different time. So here you can see the negative Q net is more risky or pro-arrhythmic. Positive Q net is safer. So evaluated all these drugs per bar graph and the plot graph. So these are the evaluation, additional evaluation from the simulation study to finding the safety index of the drugs. Okay. Now this is the last slide. So we have already know what they have done, what they have tested, and their finding. The finding to add or to recommend the SIPA that this is the safety index of this 11 drugs. In future, we can do for more drugs if we get experimental data for the particular answer. Okay. The first one is circadian infinity ion channel and arithmetics. Then IKR OD model, particularly this model, 11 arithmetic drugs and 24 dosing time. Then QNet is the safety index metric that is affecting the TDP. Then experimental data show that sodium, calcium, potassium are 1.5 into conductance in a day, the varying pattern. Then this is the most important. High risk drugs appear safer than low risk drugs depends on the time of administration. So this is the finding from this study. So if you define high risk, low risk, this does not mean that high risk is always dangerous. The low risk drugs are dangerous at a particular time. So the suggestion is, if you are taking the drugs, then consider the timing. So chronopharmacology may enhance the drug safety harassment according to SIPA standard. Okay. This is end of